Did you guys get the email that I sent with the things that you need for today?
Yeah, I got the email.
We're gonna do it. The first thing we're gonna do is a lab on cell division. Mitosis is what we call it, but we're gonna do a lab on cell division first. And then we're going to go back and finish chapter 4, because the end of chapter 4 sort of relates to cell division. There's a little side story that I want to kind of talk about, and talking about cell division first, and then going back to that makes more sense. But it's just a little section. I mean, if you look at your notes for chapter 4, it's just the last page. It says cell aging and death, and it's just a little bit of information. So we'll do that lab first, and then we'll finish up 4, and then we'll go right into the chapter 5. Grades, as far as I know, everything's been graded, and I sent you an email whenever I graded the lab test. I sent it to you, and then for the lab reports, each lab, it's a lab folder, but basically it's all the reports that you do for a particular test. Okay, so for all the labs that we've done for exam 1, I took them and graded them, and I gave you a score out of 25. So if you go into your lab, if you go into your grade book now, it'll say lab folder 1, and you'll have a score out of 25 for those labs. And so yeah, well, there were some virtual labs. There was the intro to virtual labs, lab number 1, and then there was a measurement lab number 2. And then you had 4 labs that you turned in lab reports. The measurement lab, where you measured your arm length and your height. And then you had pH, enzymes, and biological molecules. And so I took them, and I kind of treated, so there were 6 labs total, and I kind of treated each lab as being worth about 4 points, and then I figured it up and then gave you a score out of 25. So when you go into your lab, all those labs will be gone from your grade book, and they'll be replaced by a column that says or a row. I guess it'd be a row for you guys, but it'll be replaced by something that says lab folder 1. And it will be a grade out of 25. One of the things I did notice was that people did lose some points there for not turning in stuff. So they didn't turn in everything they were supposed to turn in. And those are easy points, guys. Those lab folder points, there's no reason why you shouldn't get 25 out of 25 on every single one of those, unless you just don't turn them in, or you don't answer every question that is on those lab reports. And so if you didn't get 25 out of 25, just keep that in mind, that you either didn't turn in something, or there was a question somewhere that you didn't answer. But don't leave those points on the table, because those are some of the easiest points in the class. We do the lab, you finish the lab report, you submit it, and as long as you answer every question, I mean, even if you get a question wrong, I just make a comment saying, hey, for number 3, this is the correct answer. And so you don't even lose points for getting stuff wrong. You just lose points for either not answering something or not turning it in. And so that's where, if you don't have 25 out of 25, that's what happened. All right. The lecture test scores were okay. I mean, I've seen worse, but I've also seen better. We had the same amount of people get an A as we had people who failed. And so I'm not sure what I can do with that. It'd be different if like everybody failed or everybody did good, but when you have the same amount of people, I mean, there was somebody in the class who missed 2 questions on the lecture test. That's it. Out of 100 questions, they missed 2, which is outstanding. And so I don't think the test was that bad, but people didn't do very well on it. And I thought you had a lot of stuff to help too. You had the study mate activities, you had Kahoot games, you had I gave you practice quizzes that had questions straight from, that I just moved to the test, including that cell respiration one where I just moved all the questions straight from that practice quiz to the test. I didn't change anything. And so, if you didn't do very well, we got to figure out why you didn't do very well. If it was a not preparing type deal, then I can't really do much about that. I can't come to your house and make you take this class seriously. You know what I mean? And if you're not taking this class seriously, it will win. It will defeat you. Okay. Because this class it's a lot of information, and you got to stay on top of things. And so if you're not, then my advice, if you're not going to take the class seriously, then maybe you should drop it. Okay. And try again later, because it requires your attention. It requires you to spend time studying. Okay. Now the lab tests were very, very good, and so that kind of balanced it out a little bit in a way. But the lecture test is worth more than the lab test. But the lab tests were really, really good. Had a lot of good grades. I think, what was that right there? 5, 10, 15, 17 out of 21 people who took it. 17 out of 21 got either an A or a B. And so that is awesome. Okay. And so you take those two things together, and all the other stuff we've done, we had a lecture test, we had a lab test, you had a lab folder worth 25 points. There was a homework assignment based on the measurement lab, which once again, a lot of people didn't submit. They didn't turn it in. They didn't even do it. In my mind as a teacher, I'm like, well, they didn't even do it because it wasn't even submitted. Then you had the pH quiz, and there was another 10-point assignment. I just can't think of it off the top of my head. Then you had the soul certificate, and then you had the student information sheet. What was that other 10 points? There was a pH quiz, there was homework, and there was something else. Let me
that little quiz that we did in class.
Yeah, no. Oh yeah, that was it. That was it. The characteristics of living things quiz. Yep. So that's the things we've done so far. So far up until today, there's 365 points possible in the class. So what you do is you go to your grade book. Okay, now the points possible in the grade book will be different because I've added stuff from for what we're doing right now. But if you go to your grade book and take your points divided by 365, that will give you your grade for right now.
I have a question.
Like I said, out of almost half the class right now has at least an A, B, or C. Actually, more than that. 70% of the class has at least a C. Now, if you're in that 30%, then it's time to wake up and start. You dug yourself a little bit of a hole, so you need to spend some time digging yourself back out of it again. It's not the end of the world though, because tests aren't the end of the world. There are going to be plenty of extra points, other points that you can get. But this whole not turning in stuff, I can't do anything about that. You got to turn in the stuff when it's due. Period. And so keep that in mind. I saw somebody who didn't start the test. Okay, didn't even start to take the test until 11:50 on Tuesday night. 10 minutes or 9 minutes before it's due. I mean, come on, guys. Okay, but they didn't even start the test until 11:50, and it was due 11:59. And so you had a whole week to take that test. And so you should have had plenty of time to find a little bit of time. I also noticed that people were finishing the test really quickly, but not doing well. And so if you have 80 minutes, but I'd see some people like finish the test in 20 minutes and get an F. And I'm like, why'd you go through it so fast? Usually if you have people who finish a test extremely quickly and don't do very well, then that means that they just didn't have a chance. They just went through it and went, I'll just answer this, and cross my fingers. So, will we still be receiving a separate grade for lab? No. The lecture test and the lab test, you just get one grade for this class. Okay, that's it, one grade. So it all goes into one big pot of points. So if you didn't do very well on the lecture test, but you did well on the lab test, then that's good. That at least evens out a little bit. But ideally, you would want to do good on the lecture test because it's worth more. All right. So, but I'll be doing it again for the second test. I'll be doing the study mate activities. I'll be doing the practice quizzes. We'll be playing a Kahoot game. All that again. And so, like I said, maybe you didn't do the Kahoot game as many times as you should, or do the study mate activities as many times as you should, or read over the notes. I have no clue. Like I said, I had the same. Yeah, I had more people doing well than not doing well, so I'll take that as a win as a teacher. And if you did really well, and you got a good grade in the class right now, then keep doing what you're doing. It's working. The Kahoot game points, though, I have not added those yet because I wanted you to see what you actually got on the test. Okay, so I wanted to see. I wanted you to see have an accurate idea of just how you did. Now later on tonight, I'll be adding however many Kahoot points you got. I'll be adding the points to your exam scores. I'll just pick one and put it in there. And I usually pick the one that I feel like needs it more. But I'll add your Kahoot points to your exam scores, and I'll shoot you an email saying, hey, I have you down as having this many points for the Kahoot game, and here's where I added it in the grade book. Any questions? All right. Let's see. Let's go into the labs. First two labs here, diffusion osmosis, microscopy. Those are labs you'll do in your own time. Okay. Now we talked about diffusion osmosis last class, so Tuesday. We talked about diffusion osmosis, so you can read the introduction there, and then down towards the bottom, there are 4 virtual labs that you do. Now this instructor demonstration videos, that's me recording myself doing the lab and talking to you about what you need to do. So you can see that each of those labs takes maybe 5 to 10 minutes, so that they're not too long. I was able to finish them. I was even explaining. I was explaining it as I go along, and so it only took me 5 to 10 minutes to do those 4 labs, and so finish those. Now this naked egg osmosis thing, it's just extra stuff. So if you want to do a little experiment at home using osmosis, then you can, or if you want to do one with your kids, or if your kid has a science project or have something they need to do for science, then you can do a naked egg experiment where you use vinegar to dissolve the shell of an egg, and then put the egg into different solutions and watch to see whether it will take in water and swell up, or whether it will the water gets pulled out and it will shrink. So it kind of mimics what happens to cells when cells are either in an isotonic environment or a hypertonic environment or a hypo. So that's just extra stuff. But now it does take about 2 to 3 days because you have to soak it in the vinegar for a while and get that shell dissolved away, and then put them in. You got to wait for osmosis to happen. But it's a nice little fun experiment you can do at home with like 3 eggs, some vinegar, and you can use maybe salt or sugar as your solute. Some people use corn syrup, which is corn syrup is what actually is used in your virtual lab for osmosis. This one that says movement of water across the semi-permeable membrane. And there's the protocol right there. So you kind of go to that protocol and it talks to you about how you perform the experiment. So here it wants you to use some corn syrup. But like I said, you can create different solutions of sugar water or salt water, because a solute's a solute. All you need is a solute that will help you to pull water one way or another. All right, so that's just extra. Go back here. Lab number 2 is microscopes. Same deal. You've got some opening information, and then you have 2, and then here's me performing the experiment. Now the first video I made is about 14 minutes long, because I explained like how, the basics of microscope before I actually started doing the lab. And so that's why that one's a little bit longer. But once again, neither one of those takes more than 5 to 10 minutes once you know what you're doing. Now, what a lot of people do is they'll just they'll do the virtual lab, and if they run into an issue or a problem, then they'll go back and watch the video to see what they're doing wrong. Okay, and a couple of you guys know what I'm talking about. A few people earlier, like a week ago or so, they emailed me and said, "Hey, I'm having trouble with this lab," and I said, "Watch the video, that I made." It was actually my biology 202 class, but I'll just say watch the video, and you'll see where what I did at that particular point. So there are some areas on virtual labs where you might get stuck, and that's why I do these videos, so you can go in and watch the video and figure out what it is that you're doing wrong. And so there you go. Those 2 labs, I put a due date of March 28th, so you've got plenty of time. Okay. However, get it done, get it out of the way. You know what I mean? Because what you don't want to do is wait until, because you're going to have a lot of stuff that's going to be due March 28th. That's the day. That's around the time I expect us to be taking the next test. Okay, or the next test will be due around March 28th. And so get some stuff done now, so that way there's less to do later. You can focus more on the test. So if you actually somebody in class have already knocked out all these virtual labs. So I guess they did it, after class on Tuesday. They got it done, So it's done. It's one less thing to worry about. And so that's what you want to do. You want to stay on top of the class. And if something, don't let everything start to pile up on you, because it does get kind of overwhelming as you get closer to the test. So get it done when you can get it done. Same thing about today. We're going to do this mitosis lab. Once we do the mitosis lab, do the lab, complete the lab report, and submit it, and be done with it. And it's one less thing to worry about later. You also have your study guide for the lab exam already up, so you can pull that up, and it helps you to kind of go through each lab and figure out what it is exactly that you need to know for each lab. So there's the stuff, once you do your diffusion and osmosis lab, or you can print this off and have it with you as you do those virtual labs, and kind of make little notes as you as you go through. And so here's the stuff, like for example, today we're doing lab number 3. And so you need to know this information, that human somatic cells are diploid. That means they have pairs of chromosomes, and you kind of work your way down. So let's look at what you need to know for mitosis. Mitosis is the official term for cell division. And that's how most of the cells in your body are produced, is one cell will copy all of its DNA, because DNA is the instruction book that drives the functioning of the cell. And that reminds me too, remember I gave you guys that extra credit assignment, that transcription translation, where I give you the DNA code, you convert it into RNA, and then you use that codon sheet and you put the letter and it spells out a message, right? That extra credit assignment that I showed you at the very end of class on Tuesday. Somebody's already finished it. They're on the ball. They're just like, I'm gonna get done. I'm gonna get done now. 10 points, boom, 10 points extra credit. And so don't let it pile up, okay? Get as much done. There's nothing due Sunday. There's nothing due this weekend. So use this weekend to get stuff done that is due the following weekend, or the following weekend. Get done whatever you can, whenever you can, because you don't want to pile up on there. But the cell cycle basically is the life of a cell from the moment it is created through cell division until it itself divides again. Okay, so it sort of starts from the time it's formed until the time it divides, and it's a continual cycle. Cells are constantly, growing, dividing, growing, dividing, growing, dividing, because there's a maximum size that cells reach where if they grow any bigger, if you ever thought about why are we made up of trillions and trillions of very small cells versus why aren't we made of like 200 big cells? It's because as cells get bigger, they actually become less efficient. Staying small is actually good. It allows you to easily and quickly move things in and out. And that's one of the housekeeping functions, right, of a cell, is bring stuff in like nutrients and oxygen and things like that. They got to do that quickly, and they got to do it pretty much continuously. And they have to get rid of stuff like waste products. And staying small allows you to do it faster. Okay, as cells get bigger, they become less efficient at moving things in and moving things out. So cells reach a point of like maximum size where instead of growing more, what will happen is that cell will just divide into two smaller cells, and then those guys will grow to a certain point, and it just keeps on going and keeps on going and keeps on going. Now, all these cells in the body, okay, other than sperm and egg are special cells that are designed to unite and create another human being. But all the other cells in your body are what are called somatic cells. Somatic, soma means body. So somatic cells mean cells of the body or cells that form the body. So all the other cells that we talk about in this class, this semester, next semester, all of those except for sperm and egg, all of those are called somatic cells. Now, every cell, and I mentioned this last on Tuesday, every cell in the body has the full instruction book to function correctly. So, there's 46 in a somatic cell. There's 46 chromosomes. Chromosomes are the structures that house the DNA. So you can kind of look at like DNA is like the information, and then imagine that chromosomes are like the encyclopedia books. Each one holds some information, and they number them 1 through 23, starting with the largest chromosome that they identified. They designated as chromosome 1, and then the next largest was chromosome 2, and the next largest was chromosome 3. But they found 23 individual discrete chromosomes, but we have 2 copies of each chromosome because sperm brings 1 copy and egg brings 1 copy, and when they unite together, you end up with 2 copies. So the proper instruction book for a human cell is 46 chromosomes, 23 pairs. So if you looked inside your cells, you would have 46 total chromosomes. You would have 2 copies of chromosome 1. You got 1 of those from your mom, 1 of those from your dad. You'll have 2 copies of chromosome 2, 1 from your mom, 1 from your dad, and then 2 copies of chromosome 3 all the way down the line. Now at the very end, the 23rd pair is what we call the sex chromosomes, which determine whether you're male or female. If you get an X from your mom and an X from your dad, you're female genetically. And if you get an X from your mom and a Y from your dad, then you would be male, okay? Anytime you have pairs of chromosomes, they call that diploid. Di means 2, okay? So all the cells in your body, except for sperm and egg, depending on if you're male or female, are going to have 46 chromosomes, 23 pairs, and we call them diploid. Now, if a cell is going to divide and make another cell, then he's got to make sure that each of the 2 cells that are going to be produced by 1 dividing, each one of those has to have the full instruction book. And so that's what it talks about here, that you know, a lot of what you see with mitosis is you're watching genetic information be divvied up between the two cells that are going to be formed. The cell that you start with is called the parent cell, okay? And then the two cells that are created from that one cell dividing, that's going to be called daughter cells. Each daughter cell has to have the full book. So before a cell can divide, the first thing it has to do is copy all of its DNA. You know, we mentioned the other day about how you might copy a segment of DNA and use it to make a protein. That's called a gene, okay? But when you go to divide, when a cell goes to divide, it's got to copy all of it, all 46 chromosomes, the entire book. And so that way each daughter cell has the full instruction book. Now, you kind of want to look at mitosis as a cell cloning itself, because those two daughter cells that are produced are exactly like the parent. It'd be like you splitting into two and, you know, creating another Chuck or another Caleb or Jeremy or whatever. You know, a clone is an exact copy of the original, right? Well, that's kind of what daughter cells are, because, you know, the parent cell is going to copy all of its DNA. It's going to, you know, now it has two copies. It's going to send one copy with one daughter cell and send one copy with the other. And those two daughter cells will be genetically identical to the parent. And they'll be genetically identical to each other. And so essentially, it's like a cell is cloning itself, creating an exact copy of the parent. And that put there some definitions. Somatic cells are any cell other than reproductive, other than sperm or egg. Diploid means you have pairs of chromosomes. Now, why would mitosis occur in the body? Happens all the time. It happens when you are developing in the womb. You know, you go from a single cell. When sperm fertilizes egg, that's a single cell called a zygote. And then 9 months later, you're a fully, you know, functional human body that's made up of trillions of cells. So a lot of mitosis happens in the womb when a baby is developing. To grow tissue, you got to do some mitosis. And if you lose cells, you know, if cells become damaged, destroyed, old, worn out, whatever, then they have to be replaced with new, fresh cells. If you have to do tissue repair, if you get a cut, you got to grow skin to seal that cut. And so any kind of injury, any kind of death to a cell, all that will trigger mitosis to happen, okay? Now, what's interesting, and people don't think about this a lot, but cells aren't always dividing. Usually a cell is just being a cell, and it's in between divisions. That's called interphase. Okay, interphase is between divisions. If you see INTER in any science word, INTER, that means between. For example, interface. Or and you're going to see, actually you see INTER in a lot of structured names in the body. If something is between two things, it'll be called the inter whatever. INTER is between. So a cell could be between divisions. Then something, and you can think of like interphase. Interphase is like a cell being a cell. It's just doing its job. It's performing whatever functions it's supposed to. It's being a cell. Then something will trigger it to divide. Like it might be, you know, injury to the body, or it might be other cell. You know, a cell's getting ready to die. You know, one cell dies, so you got to replace it with a new fresh cell. That happens a lot. But something will trigger it to divide, and when it's triggered to divide, then it will start what we call the mitotic phase. There's a PowerPoint down towards the end that we're going to kind of look at first, and then we can come back and look at all this stuff right here. So there's a PowerPoint down here that has some of the basic information about mitosis right there. So it says mitosis is cell division that occurs in somatic cells. Meiosis, which is another form of cell division, that's the cell division that produces sperm and egg. And that's covered next semester when you get to the reproductive system. So we're talking about the most common form of cell division that happens all the time in your body, mitosis. One cell is going to divide and produce two, and it's necessary for development, tissue growth, replacement of old or dead cells, and tissue repair. The cell cycle is divided into two major phases. Inner phase, where a cell is just doing its job, being a cell. And that's actually where a cell spends most of its time. A cell spends most of its time in inner phase. It's not actively dividing. And usually whenever a cell is triggered to divide, it does it quickly, usually within about an hour. And then it goes back, those two daughter cells that are produced will go back into inner phase and do their job, whatever the job happens to be. So a cell's just doing its job, then it's triggered to divide. It'll divide quickly, and those two cells will go back to doing their job, whatever their job might be. And so here it kind of talks about that, a cell spends most of its time in inner phase, which is the blue. But what they'll do is they'll divide interphase up into three sort of subphases. You got what's called a G phase, that's called G1, and that's called G2 over here. And what that is growth. And essentially, the cell is growing, and it's performing its function. But there is a subphase right here that we need to be aware of. So this happens during interphase. Prior to a cell dividing, I mentioned this a minute ago, prior to the cell dividing, the first thing it has to do is copy all of its DNA, so that way each daughter cell gets a copy. And so that's what happens here. This is called S phase. And the S stands for synthesis. And during that phase, that's when a cell copies all of its DNA. So the cell is going to start out with 46 chromosomes. During S phase, it makes a copy of the entire book. And then it's going to send one copy into this cell and send the other copy into that cell. And when you look at a cell dividing underneath the microscope, that's what you're seeing. You're seeing the genetic information get divvied up. After the cell copies it all, then he's got to sort of make sure that each cell gets the full instruction book, so you're watching the divvying up of the genetic material. That's what's happening down here during the actual mitotic phase. It's just the divvying up of chromosomes, making sure that each daughter cell gets 46. Any questions? So essentially, whenever a cell's created, it'll enter interphase, it will grow, and it will do its job. Then when it's triggered to divide, it will copy all of its DNA. Then it will grow a little bit longer, sort of prepare for division, and then it will divide. Then the two daughter cells that are produced go back into the cycle and repeat it however many times it has to happen. There's interphase. The cell is between divisions, just being a cell, doing its job. Now the DNA, when a cell's not actively dividing, you can't see the DNA underneath the microscope. The only time you can see the chromosomes is when a cell is actually in the process of dividing, because usually DNA strands, chromosomes are very long and sort of stringy. You got your G1 phase, which is part of interphase. You're growing, you're producing things. And S phase, you're going to copy all of its DNA. It's going to grow for a little bit longer. It's a brief phase there, G2, and then it will enter into division. Now, if you're looking at a cell who's not actively dividing, it'll look like a typical cell. You'll be able to see the nucleus, after you stain. Remember, you always got to sort of stain to get contrast with a tissue. So if you would take a cell and stain it, the nucleus would just look like a dark blue spot. if you're using a methylene blue, which is a stain that's used a lot to stain cells because methylene blue will stain the nucleus. But you're looking at there, just looks like a typical cell. You can't see anything interesting about it. It's just a cell, okay? Because it's in interphase. It's just being a cell. Now, when it does start to divide, you can see the division process. You can see the mitotic process as you watch the chromosomes get divvied up. But you can't really see the chromosomes in interphase because they're really long and thread-like. But what happens during mitosis is the chromosomes basically shrink down but get fatter. They condense down. It's kind of like the cell's packing its bag so it can easily move the material from one cell to another. Whenever the cell begins to divide, they call it the mitotic phase. And it's basically 4 steps and an event, okay? The 4 steps are called prophase, metaphase, anaphase, and telo or telophase. People pronounce that different ways. I always learn telo, so you might hear me say telophase a lot, and you might hear another teacher say telophase. I try to say them both, but after a while you're like, you know, telo or telo, telo or telo, telo or telo. So I usually call it telophase just because that's how I always remember it from my teacher telling me. Was you trying to say something, Kirsten? There's a yellow there's yellow around your box.
No, my dog stepped on my computer.
Okay. All right, so prophase. Okay, here's where you start to see the chromosomes. The chromosomes are usually long and thread-like, so you can't see them during interphase. But a couple of things happens during prophase. So this is where the cell is starting to divide. This is the first stage. Now, pro can mean before. Pro can mean first, like prototype, you know, the first type, that sort of thing. Pro has a lot of different meanings as a prefix. But here you can kind of look at it as, you know, the first phase is prophase, the first one. And what happens is the cell basically starts the division process. The first things that has to happen is the chromosomes are going to condense down and become shorter and fatter. And I kind of look at it like the cell is packing its bags so that it can move the DNA a lot easier. This is really no different than if you're going on a trip. You know, you don't just take your clothes and throw them into the back of the car. You pack them into an easily transportable or portable, suitcase or something that's easy to move. So that way, and that's what's going on here. By condensing and becoming shorter and fatter, it makes it a lot easier to move the chromosomes to where they need to go. So this is just like a cell packing its bags, getting ready to move the DNA. Also, the nucleus, because the nucleus is holding the DNA, for a brief amount of time, we need that nucleus to break down. So that way we can move the chromosomes. Now, once the chromosomes get moved and the cell divides, then the nucleus will reform and repackage the DNA. Now, this whole idea right here of there is a moment in time during division where the DNA is not inside the nucleus. That will actually come into play a little bit later when we talk about cell aging and death. But the nucleus is going to break down, and the chromosomes will start to collect towards the middle of the cell, and kind of looks like worms in the middle of a cell, especially on the picture you're going to look at here in a minute. You can sort of tell it's prophase because it looks like there's these little worms collecting towards the middle. And then what's cool is you go into what's called metaphase, where the chromosomes basically line up in the middle of the cell. It's an incredible thing. This is the second stage of mitosis, metaphase. All the chromosomes will line up in the middle, and you'll get the production, okay? These right here are called centrioles. They're important for cell division. These are structures that are formed for cell division, and then they get broken down again. So the only time you really see them, or the only time they really have a job, is when the cell is dividing. But what these structures will do is they sort of migrate to two sides of the cell. Because if you look at it here, let's go back to this first picture here. The centrioles are Well, it didn't really give it to you, but you can kind of see the centrioles starting to make their way towards the opposite sides of the cell. And then the centrioles will send out these protein fibers, and you can see them here. Okay, these are called spindle fibers because they resemble a spindle in an old-time sewing machine. So I don't know if you've ever seen an old-time sewing machine, but a spindle is like the threads are pulled real tight, and so they're real tight and thin. So it kind of resembles a spindle, and so they call those spindle fibers. You also hear it called the mitotic spindle. Okay, but essentially these protein fibers are growing out this way from those structures called centrioles. And what those spindle fibers will do is they'll attach to a chromosome. And you can see the spindle fibers a lot of times. If you look just beside the chromosome, you can see these little thread-like things kind of coming out like that right there. Metaphase is very easy to recognize because the chromosomes, which stain a purple color or a dark color, the chromosomes all are lined up in the middle, and you'll see this dark line right down the middle of the cell. So if you look at a cell and you see a dark line right down the middle, that's metaphase. Meta in this case means between. So they line up between, in the middle. So meta, you can kind of look at meta as meaning middle. It means between, middle. And the spindle fibers grow out. So you got your centrioles there, spindle fibers grow out, and they'll attach to the chromosomes, and that's metaphase. Then these protein fibers will contract. The word contract basically means they will shorten up. And as they contract, they pull the chromosomes towards one end of the cell. And so what you get is you get the chromosomes, they will separate from each other. So all the copies, the copy of chromosome 1 that they'll get pulled apart. And so the chromosomes get separated. And when that happens, that's called anaphase. And when that occurs, you'll actually see two lines in the cell, in the middle of the cell, as the chromosomes start to get separated. They almost kind of it's kind of like when somebody got jerked from behind and their arms and their legs would move one way. It kind of looks like a person getting jerked from behind with the chromosomes sort of taking on an appearance like this right here that you see on this picture. The chromosome will kind of look like that with a spindle fiber pulling it that way, just like if somebody got jerked from behind, their arms and their legs will kind of fly forward as they get pulled backwards. Kind of what it looks like to me. That's called anaphase. Ana, okay, that word means apart. So anaphase is the phase where the chromosomes get pulled apart. They're going to get pulled towards the opposite sides. So they're going to come this way. And then once they arrive over here at the opposite ends of the cell, then the cell will start to split down the middle. Okay. You'll get these invaginations occur. They're called cleavage furrows. But essentially, you'll start to see the cell pinch into two. And a lot of times, if you see a cell in telophase, it kind of looks like a peanut. It's in the process of squeezing like that, and then eventually separating into two. So once you get those chromosomes pulled apart far enough, the cell will start to pinch in, and then all it needs to do now is separate. The chromosomes arrive at opposite poles. The nucleus will start to reform, and the cell will start to pinch itself and divide into two. The word telo or telo means at the end of something. All right. And so also telo can also mean distance, like they're far apart from each other. But in this case, it basically means at the end. That means last stage. Okay. And all the cell has to do is continue to pinch. So he'll continue to pinch down the middle until eventually you have two cells, and those two cells will separate, and now you have two. It's not a stage when the cell the event of cell separating. It's not a stage. It's an event, and it's called cytokinesis. So cytokinesis is, the first the mitotic phase is sort of like the divvying up of genetic information, and then cytokinesis is sort of like the divvying up of the other internal components, the cytoplasm, making sure that each daughter cell also gets some cytoplasm with some of your organelles in them, like your mitochondria, your ribosomes, some of those organelles that we talked about earlier in chapter 4. This cell down here at the bottom is almost done. All it has to do is pinch off that last little bit, and then it will separate out into 2 strands. So that's kind of mitosis. Let's go back to the lab and look at cells that are actually in the process of mitosis. So it says here, in this lab, we will explore phases and events of the cell cycle. We will primarily use the embryonic stage of a whitefish. Okay, a whitefish is a fish. And a lot of mitosis happens very early in development, where you go from one cell, and then it will divide into 2, then 2 will divide into 4, and 4 will divide into 16, or 8, and then 8 will divide into 16. And so very, very early in development, organisms are basically a ball of cells. And so that's what we're looking at. We're looking at the very, very early stages of a fish, all right? But even us, if you would look at a human very early in development, we're talking within, a couple hours or days of development, it's going to be a ball of cells, because it starts out as 1, then that cell splits into 2, and those 2 guys split into 4, and those 4 guys split into 8, and you get this ball of cells. It kind of looks like a blackberry. All right, that right there is a rapidly dividing baby, basically. And so when they say embryonic stage, it's basically a fish baby, very, very, very early. And so it says at this stage, the organism is a ball of cells undergoing rapid division. And so when you take a snapshot of a ball of cells, most of those cells are just, in interphase. They're not doing anything, but every once in a while, you'll find a cell that is in one of the stages of mitosis, and you can use it to look at it. So we actually use whitefish slides. They're kind of like one of the go-to slides to teach mitosis with animal cells. It says, while completing the lab, you can refer to the PowerPoint. This right here breaks it down again. You spend most of your time in interphase, growing, performing the job. Then when you are triggered to divide, you're going to copy all of your DNA. That happens right here during S phase. And then once he's copied all of his DNA, he'll enter a brief growth period where he prepares for division. And then he will go into mitosis, divvy up the DNA, and then divide into 2. Once you get those 2 daughter cells, then they go back into interphase and continue to do their job. So this is a fish, baby fish slide, a fish blastodisc, a whitefish blastula. But it's a ball of cells, and if you notice there, most of the cells are just cells. You don't see any obvious things going on. But then you see every once in a while, you'll see cells that are in some of the stages of mitosis. Right there. All right, the chromosomes are those dark little structures. Now, the reason they call them chromosomes is because chromo means collar, and soma means body. So they look like they're the collared part of a cell when you're studying mitosis, chromosomes. That's where they get their name from. So you're looking at these cells and you're trying to figure out like what stage you're in. Like this guy here with the dark line down the middle, that's metaphase. That's where the chromosomes line up in the middle of the cell. This one looks like little worms in the middle of the cell. That's prophase. So what you want to do for the test is if I show you a picture of a cell in a stage of mitosis, you can tell which stage it is. So let's kind of look at each individual stage. All right, interphase, you can't tell anything obvious. It's the period of time between divisions. The cell's being a cell, doing its job. It's growing, it's copying its genetic material, and it's preparing for division. A typical cell spends most of its life in, most of the cell cycle in interphase. When it's triggered to divide, it does it quickly, and then it goes back into interphase. So interphase is between divisions. You can't see the chromosomes. When you look at a cell underneath the microscope, it just looks like a typical cell. You'll be able to see the nucleus, but you can't see the individual chromosomes. And then there's the three subphases there. And it says, like I said, none of these events occurring during interphase are visible under a light microscope because the chromosomes are loosely coiled. They're not packed together. They're not condensed. And so it just looks like a cell in a random tissue sample. So there's the interphase, okay? Then it goes into the different stages of mitosis. When triggered to divide, a cell will enter the mitotic phase. During this phase, the chromosomes will condense, become short and fat, and that's when you can start seeing them with the microscope. This kind of makes them easier to move, and then you can use the chromosomes, like where's the chromosomes at, and all that to determine the stages. So prophase is first. It's the first stage of mitosis. During this phase, the nucleus is going to break down. The chromosomes will start to condense and become visible. And the centriomes, or centrosomes, also called centrioles, but the centrosomes will move towards the opposite ends of the cell, and they'll send out their spindle fibers. And then they will, that way they can divvy up the chromosomes and move them to opposite sides of the cell. Now you won't see that. All you'll really see is you'll see the chromosomes start to collect towards the middle. And so you'll start to see the dark spot, but there's really no definition to it. It's just a bunch of chromosomes collecting towards the middle. And sometimes it looks like there's little black worms in the middle of the cell. So that's prophase. The nucleus is broken down, chromosomes are gathering towards the middle, chromosomes are condensing, they're packing their bags, and they're getting ready to be moved. Then all those chromosomes will line up in the middle, and then you're on to metaphase. Metaphase, the spindle fibers will grow out and grab a hold of them. And sometimes you can actually see the spindle fibers like you can in this picture here. You can see the little thread-like spindle fibers attaching to the chromosomes right there. Now this cell is in prophase as the chromosomes are collecting towards the middle. And it looks like this guy down here is also in prophase. As the chromosomes are starting to condense towards the middle of the cell. But metaphase, if you see that dark line down the middle, that's metaphase. There's a nice little picture right there as well of the chromosomes lined up. Once the chromosomes get pulled apart, you're going to see two dark lines, and that's called anaphase. And so you can see a picture there. The cells are getting pulled to opposite sides. And ana means apart. So if you're looking for like a key word in a question, if you see chromosomes are pulled apart, if you know that ana means apart, then that's anaphase. That's where it gets its name from, because the chromosomes are being pulled apart, anaphase. And then there's a picture of it there. And once again, you have a cell up here in the corner that's in prophase. So this one up here in the top corner, that one is beginning the process of mitosis, while this guy is moving towards the end. He's pulling the chromosomes apart. And then all he has to do is get these chromosomes over to about right here, and then he can start to split down the middle and form two daughter cells. And then telophase or telophase, that's when the chromosomes arrive at the opposite ends, and the cell will start to pinch itself off, and it kind of looks like a peanut in a lot of cases. In fact, if you see that little line right there, you can actually see the cleavage furrow, the little line that develops. And so you can see that this cell is just about done. Okay, all he has to do is just finish that pinching off right here, and he will be done with cell division. So he's just about finished. You can see the peanut shape there. If you follow it around, kind of has that pinching off in the middle, the cleavage furrow. Looks like a little peanut. That's telophase, or you can call it telophase. Same thing here. Kind of has that peanut shape to it. The two dark lines, the two lines of chromosomes are kind of far away from each other, and he's basically in the process of pinching off. Now these are microscope pictures using a standard light microscope, and the advancements in technology that we have nowadays has really opened up the ability to essentially watch mitosis occur in real time. Right? So they actually have recordings now where you can watch a cell as it's in the process of cell division. Cytokinesis is the cell basically splitting into two. It's not a stage of mitosis, it's basically an event as it pinches off and turns into two. So cytokinesis basically happens at the same time as the last stages of mitosis. You might see it start to pinch, you might see the cells start to pinch off during anaphase or during telophase, during the last phase. As it's, once the chromosomes get pulled apart enough of a distance, then that cell will start to pinch itself off. Now this right here is a picture of a bunch of cells, all in different stages. And so when you look at this right here, this guy's in metaphase because you see the dark line down the middle. It's a single line. This one's in metaphase, dark line down the middle. This guy here, you can actually see it starting to pinch. And so that's the cell right there. So this one's in telophase, the last stage. Sometimes it can be hard to distinguish because there's no real point where one stage ends and another one begins. So sometimes if they're like sort of right in between, like this right here, if you're looking at this one, I mean, he's kind of at the midline between anaphase and telophase. Yeah, the chromosomes are being pulled apart, and you are seeing a little bit of a pinching going on there. So he could be anaphase. You can describe that as anaphase, or you can describe it as telophase, and you're kind of correct. When I choose pictures for the test, I don't want there to be any confusion on what stage you're looking at. And so that right there is kind of like, we can call this late anaphase or early telophase. It's sort of like right there at the junction between the two. Same thing with this guy. That looks like anaphase, but he's also very close to being in telophase. Maybe if he moves a little bit more over here, then the cell will start to split down the middle. So sometimes it can be hard to tell which stage you are in if it's right there at the straddle point. Metaphase, that's a beautiful metaphase picture right there in the middle. Dark line, you can actually see the spindle fibers right there growing out. That's a very pretty picture. This guy looks like he's in metaphase. That's probably prophase, but yeah. When I choose the pictures for the test, I'm not trying to trick you. I will choose one to where there's like no doubt of what you're looking at. And so when you look at these, you kind of look at them and go, okay, it looks like this guy over here is in metaphase, right here. It has a dark line. I would call this anaphase because you can see the two dark lines as they're being pulled apart. This one looks like he's almost done. I'm trying to figure out if this is him right here. That looks like telophase because he's splitting right there down the middle. I'd say this guy's prophase because the chromosomes are starting to collect to the middle. And so what you're going to do is in your lab report is you're going to draw the different stages of mitosis. And so, you can draw them a certain way. There's some more pretty pictures. So you just practice looking at them. And then here we've got, advancements technology where we can add like fluorescent proteins that glow. And so we've got that going on there. And you can see, this is prophase here. C right there is metaphase. D is anaphase as it gets pulled apart. And then this right here would be like telophase and the cell splitting. And then we actually have videos now that you can watch of the cells kind of moving from one stage to the next. There's metaphase. You can see the dark chromosomes there. Here's chromosomes right here and right there. So that would be anaphase. The chromosomes are getting pulled apart, and you just happen to snap a picture. Because whenever you take a slide sample, you're kind of snapping a picture at that moment in time. So this cell here looks like, it's anaphase as the chromosomes are starting to separate from each other. You can see 2 lines of chromosomes versus 1. And then now we also have videos. That cell right there is almost done. Okay, so that cell's almost finished. All he has to do is finish up. And then you got some videos like this right here where you can watch the different stages. Now, keep in mind that the stages are continuous. It's not like the cell goes into prophase, stops and waits a little bit, then goes into metaphase, stops and waits a little bit, and then goes into anaphase and stops. When you snap that picture, whatever phase they're in is what you're going to get. But these pictures here, these videos allow you to see like all the, it's a time lapse photo, but it takes about an hour or so, maybe less than that, depending on the cell and how fast it divides. So the guy on the video on the left there, you can see metaphase, anaphase, telophase, over. So it happens very quickly if you do a time lapse, which is what's going on here. It's really cool looking though. Any questions? I can like sit and watch this all day. It's kind of mesmerizing. Got your lab report here. So let's answer these questions real quick. So if you have your lab report with you, I'm going to give you some answers. Let's see. During which phase of the cell cycle does a cell replicate its DNA? That's S phase. The S stands for synthesis. Those are all part of interphase. These 3 right here. That's when a cell's between divisions. But S phase is where a cell copies all of its DNA. How many chromosomes should a human somatic cell have? That would be 46. 23 pairs. When an organism has pairs of chromosomes, it's called diploid. DIPLOID. And all the cells in your body, except for sperm and egg, are diploid cells. So 1 is B, 2 is 46, and 3 is diploid. Are we all good with that? I got to erase it to move down. All right, no objections. Moving on. Three reasons why a cell may undergo mitosis. You can put things like, oops, sorry. There you go. Growth, tissue repair, so growth and development might be a better way to, during growth and development, tissue repair, replaced, to replace damaged or lost cells, damaged or lost cells, to replace cells that need to be replaced. So that's like 3 of the main differences: growth and development, tissue repair, and then to replace damaged or lost cells. In which of the following phases does a cell spend most of its time? That's interphase. The cell spends about 80 to 95% of the time not dividing. It's doing its job, whatever its job is. And then when it needs to divide, it does it quickly, and then it goes back into interphase. All right, moving on down. Which of the following shows the phases of mitosis in order? PMAT is what P right here. PMAT. Prophase, metaphase, anaphase, and telo or telophase. So it'd be this one right here: prophase, metaphase, anaphase, telophase. Then it has you draw so 6 is E, and then it has you draw pictures, and how detailed you want to get with this is your choice. I am not an artist. Interphase, it's just going to be a cell being a cell. You're not going to see anything fancy with that. Prophase, I would draw it looking something like little worms in the middle of the cell. Something like that. Notice the nucleus broke down, so you can move the chromosomes. Chromosomes got nice and shorter. And so I would put something like that. And then for metaphase, I would draw a cell. I would put chromosomes down the middle like that. And then maybe have your centrosomes, and they're coming out like this, sending out their spindle fibers. Maybe something like that right there. So you got your interphase, you got your prophase, you got your metaphase. Then for anaphase and telophase, for anaphase, I would do something like I'd have the cell, I would have 2 lines of chromosomes, like that. And then have these spindle fibers like over here, those spindle fibers over here. That was kind of crappy. Let me do that one a little bit differently. Have them kind of coming to a central point like that. And if you wanted to put like arrows saying, these guys are going this way and these guys are going that way, they're being pulled apart. And then telophase, I would make a little peanut. Put a dark line over here. Put a dark line over there. There you go. Something like that. Now on the test, I will take a picture of a cell and put it on the test and say which stage is this cell in. Is it in prophase, or is it in metaphase, anaphase, or telophase? And like I said before, a lot of times with these lab reports, I just move these questions over to the test. So number 6 right here, I can just move him over and say which of the following puts them in order. I could just move this over to the test. So whenever you fill out a lab report, we just kind of completed it together. And so, finish up what you need to, submit it, and then you can always print off blank copies of this and practice filling it out. But you will see these questions again on the test. So if you look at the study guide, it says cell cycle and mitosis. You need to know that human somatic cells are diploid. That means they have pairs of chromosomes. You have to know what the word diploid is, pairs of chromosomes. Somatic cells have 46 chromosomes or 23 pairs. You got reasons that cells may undergo mitosis. A cell spends most of its time in interphase. A cell copies all of its DNA during S phase of interphase. Then it kind of gives you a description of, so here's what you might do with number 6 right here on the study guide, is you may match the stage of mitosis with the description of what's happening. Okay, it'd be kind of like a little matching deal there. Somewhere, one side it will say prophase, and somewhere on the other side it will say nucleus breaks down, chromosomes condense, and stuff like that. So you might have to do a matching. And then the word that we use for when a cell splits into two, that's called cytokinesis. And then it says be able to identify a stage of mitosis. If I give you a picture of a cell and I ask you what stage is this. Good deal?
Yep.
All right. About 10 minutes left, so let's go into, before our break, let's go into chapter 4 and kind of finish up the talk about cells, cell aging and death, because it kind of ties in a little bit to mitosis. A cell only has a certain number of divisions it can do before it's done. So let's talk about that. There's a clock built in to cells. They are, they don't live forever. Now, the only cells that could live forever in the body, anyways, are stem cells and cancer cells. Okay, stem cells and cancer cells, because they do have the ability. I mean, if you continue to give them the correct environment, they do have the ability to divide infinitely, so to speak. All right, so we got a little place there with mitosis. It's basically the same information as I put on the PowerPoint. All right, so aging, it sucks, but it's a part of life, and you can't avoid it. It is what it is. But aging is just a normal, continuous process of its accumulation of damage over the course of years and years and years. Go back to chapter 3 where you had cell respiration. One of the things that happens during cell respiration is electron leak. And whenever you have electron leak, you get the production of free radicals. And free radicals damage your body. And the accumulation of that damage as you get older is one of the reasons why we age and eventually die. And so one of the processes that keeps you alive, cell respiration, the production of ATP, is also one of the reasons why you die. Same thing going on here. Cell division. You need to do cell division to stay alive. But a byproduct of cell division is what eventually kills you too. So that's kind of ironic that the two main processes that you have to do to stay alive, make ATP and cell division, are the two primary reasons why we get old and die. But what happens is, cells do accumulate damage, and they will reach a point where they are no longer functional. Now, when that happens, they will typically sort of self-destruct, and then that triggers the body to replace it with a new fresh cell. And so that does happen sometimes. Cells can actually sort of hit the self-destruct button when necessary. And then when they self-destruct, that basically triggers the body to replace it with a new fresh cell. So that's happening, okay? Now, there is a type of cell death, though, that is unplanned, okay? So you do have unplanned cell death that occurs due to trauma, infection, or other factors, lack of oxygen, stuff like that. And that's the type of cell death that you are going to see a good bit working in the medical field. That's called necrosis. Necrosis is when cells and tissues die before they're supposed to. And so you hear about like gangrene, necrotizing fasciitis, frostbite. Frostbite's a type of necrosis. And that's basically just when a cell dies before it's supposed to. But then you have a programmed cell death. And so, yes, cells do die a lot before they're supposed to, but then it's actually built into cells to self-destruct when necessary. So why would a cell self-destruct? It actually happens during development, because when we develop, we all have flaps of skin between our fingers and our toes. But the cells in those flaps of skin are supposed to self-destruct, and that tissue is supposed to fall off, and that's what gives us our grooves between our fingers and toes. And so that programmed cell death occurs during development, and sometimes that doesn't happen properly, and people are actually born with fused fingers or with webbed fingers and toes. We all start out with the webs, but those cells are supposed to self-destruct, okay? And then that's what gives us our grooves. We all have, early in development, we all have a flap of skin that connects right here from our, like right where our ear's at, down to our shoulder. And it's supposed to, those cells are supposed to undergo programmed cell death and die. And that's what gives us our narrow neck. And when that doesn't happen, then people can be born with a webbed neck. It's actually very common in a condition called Turner syndrome, which is a female chromosomal abnormality. But Turner syndrome, a lot of times the girls have a webbed neck. And so that does fail sometimes. You can see it here, where that programmed cell death does not happen the way that it's supposed to. So you got the webbed neck. It also happens in Noonan syndrome, which is another genetic abnormality. The same deal. You can end up with a webbed neck where the programmed cell death didn't occur the way that it was supposed to. You can end up with webbed toes and webbed fingers, because that's basically a failure of programmed cell death in that particular area. And so it is a part of development, just like how a tadpole loses its tail as it transitions to an adult frog. That's programmed cell death. The cells that are in the tail are going to die and slough off, essentially. Programmed cell death happens all the time. In fact, the word is apoptosis. You can also pronounce it apoptosis, apoptosis. There's different ways you can pronounce it. And really the debate is, do you or do you not pronounce the second P? Okay, that's what it's all about. Some people do. Okay, the way I learned it was apoptosis, but I think apoptosis or apoptosis, I think originally that second P was silent, because if you break that word down, apo means off or away, and then tosis with a silent P. Tosis means to fall, okay? To fall. Like when your eyelids droop, when they fall down, somebody has oculomotor nerve problems and they have that drooping eyelid, they call that ptosis. It means to fall. Well, if you put those two words together, apoptosis or apoptosis, it means to fall off or to fall away. Because whenever you have skin or tissue die, it's going to fall off, okay? Apoptosis was originally studied or described in plants, okay? Because you know how plants, like flowers, right? Easter lilies are going to come out. Easter lilies will bloom, and then those cells that form the petals and stuff, after they're done blooming, will die, and the petals will fall off. That's programmed in. That's programmed into the flower, right? Once it blooms, then it dies. It happens every year, right? Every year, the trees out here in the forest, those cells that are in the leaves die and the leaves fall off. That's programmed into the tree. That once it hits a certain temperature, once it hits a certain time of year, then the leaves will die and they'll fall off. Okay, so it's built in. Okay, we also have it built into us too. In fact, skin cells cannot play their role as skin cells unless they die. They have to die, okay, to become skin. It's programmed into them. It's a process called keratinization, and we actually talk about it next week. But this process, keratinization, that happens in skin cells, it's programmed in to skin cells to die on purpose. And if they don't, then they can't play their role as skin. And so apoptosis happens more often than you think. But in the hospital, you're going to see a lot of necrosis. Necrosis is unplanned death, and then apoptosis is actually programmed death. Another reason why cells may self-destruct is when they are old and they've accumulated so much damage that they'll just self-destruct because they are no longer useful to the body, and then the body can replace them with new fresh cells. And then you've got sometimes when cells are infected with a virus or a bacteria, they will self-destruct to try to limit the infection. They're like it's kind of like they're taking one for the team. You know, they will self-destruct to try to stop the infection from spreading to other cells. Because that's how viruses work. They hijack a cell and then use the cell to make more virus. So sometimes when a cell gets infected with the virus, it'll just blow itself up basically and take the enemy with it. Kind of reminds me of those war movies where somebody knows they're going to die, so they grab a grenade and they grab the enemy and they just blow them both up, blow the enemy up, but also blow themselves up too. So they're kind of like taking one for the team. That's sort of what apoptosis is in a way. It's a cell dying on purpose for various reasons. Necrosis, though, we actually talk a little bit more about necrosis at the end of the next chapter, chapter 5. So cells are going to die. Okay, cells reach a point where they are done. It's just when you're old, you know, when you're 70, 80, 90, 100 years old, you have a lot of cells that are all sort of dying at the same time due to the accumulation of damage, due to the loss of the structures that we're going to talk about when we get back from break, which has to do with cell division. There's a built-in number of divisions that a cell can do before it's done. And so when we get back from break, we'll finish up talking about chapter 4. And then whenever we finish that, we're going to go right into chapter 5. So have your chapter 5 notes ready. We'll finish up this talk, and then we'll go right into chapter 5 and talk about tissues. Take a 10-minute break and come back in.